stretching my leg, I promise. Donkeys power people from the southern tip of Africa to the eastern Ethiopian, East Africa, which is the country of Ethiopia, the most northern one. But we know that donkeys power people all over the world. And their ability to support, and we've had some fantastic presentations that have dealt with some of the facts and figures regarding the impact that donkeys have made in the world and are helping build the economic power of communities, whether it's women, men, communities, transport, etc. About 10 years ago, an excellent economic study was done by the International Livestock Research Institute in Kenya. And that study, I was fortunate to be part of it, but what was the question being asked was, what constrains the animal health of animals owned by the poor, resource poor? And when they asked the question about donkeys, there were three answers. The top of the charts were parasites, trypanosome parasites, number 2, and number 3, wounds and injuries. And so we are interested when we talk donkey health, we are of course included. As I was getting ready for this presentation and looking at donkey parasites, and the presentation is entitled Donkey Parasites, Detect, Diagnose, Delete. And we have to, my husband Eddie attended last year as well, we have to thank you for the feedback we had at the meeting since then, through Eric, through the Center for Professional Education, so that we could get a handle on what we really needed for parasites. And everything that's happening today in the lab has been restructured based on that. But I also found myself sitting back as I prepared for this and asking, what do we actually know about donkey parasites? How much has really been published? How much are we extrapolating? How much from our horse knowledge? So I did a quick PubMed search, which is really looking at the scientific literature over the last 50 years. And I was reminded that we've published more than 4,000 papers on donkey parasites in horses, but only not even 300 in donkeys. Now, this is a very quick look, and I'm interested in taking this to the next stage with the colleagues who've helped. You're a great professor of library science at the University of Texas A&M. But the point is that although we're going, and we've done it through the whole symposium, we're going to talk about donkey parasites, we are extrapolating. I and others have experience. Some of the anecdotal observations we make, I'll try to bring into the session now and later as to what those differences are, and there may be folks in the audience who will add to that, I'm sure. But I was amazed to be reminded of really how little we have actually published. So, where do you come in? Every one of you has an individual donkey operation. It may be a farm, it may be a premise that's smaller, it may be a very small area that has limited grazing. But every single person in this room has the opportunity to become a mini researcher. And the tools that we're going to talk about today and monitoring what's going on your premise and your operation can make a difference to the database that we have. We'll talk more about that as we move along. One of the persons I mentioned last year and I bring up because she is still one of the gold standards is Dr. Gertrude Tyler, who did a study in South Africa on the strongwell parasites of donkeys, mules, horses, and zebras. And her study is, to this day, still one of the as complete as you can get in one publication. And we continue to draw upon that knowledge base globally for what we are telling you and talking about. If we were to peer in at this very moment into the intestinal tract of a donkey that was not treated and was on free-range grazing perhaps, we would see the presence of many, many parasites. Many, many parasites. These are the larger ones, and when you start looking, you'll see the smaller ones. In addition to large numbers that our equines can tolerate or manage, numbers of worms, we also have more than 100 worm species. From a scientific standpoint, this is an extraordinarily fascinating animal to have studied my whole career as far as equids, because of the wide biodiversity that we know they have. Okay, where are we going today? We have 3 questions on our roadmap. What parasites are detectable? What parasites can we diagnose? And what are the best practices for deleting those parasites? We're going to talk about the ascarids, which are the roundworms. We're going to talk about the strongvalves as a group, and we're going to talk about the tapeworms. And we're focusing on those 3 groups, not because they're the only, but because those are the 3 most common in the horse, probably most common in what we know with donkeys and other animals. Currently today in North America. We're talking endoparasites. We will not talk about ectoparasites. In lab, we have examples of ticks and flies, and we're delighted to talk to you as much as you want to hear, but we will be focusing on endoparasites for today's symposium. This just reminds us of the diversity of the anatomic sites in a donkey, outside and in, as far as, and this is not a new organ site. It's making me hungry. That's a K that got away. This is used to just remind us of how diverse the anatomic sites are, internal and external, in our equine host, the donkey, and every other equine. Okay, let's talk the ascarids. These are the roundworms. We usually associate them with young animals. They will invade the small intestine, liver, and lungs. So let's begin with the stage that is ingested during grazing, which is the egg stage with an infected larva inside. And once that ascarid, we talk about them as parascarids, you'll hear me call them roundworm ascarids interchangeably. Once those are ingested, they will migrate through the liver and lungs and then return to the small intestine, where we'll see adult male and females eventually grow to sexual maturity. They will copulate and eggs will be cast in the feces, and that's where the animal becomes infected. Now the roundworm egg, this egg is very thick-walled, and as a result, it can survive in the environment for up to 10 years. In horses, we see an age immunity where the foal does not, eventually by 12, 18 months of age, it's rare to see parascarids present in horses. But we've often seen, the same in donkeys, but we see older donkeys are sometimes infected. And this was, I first saw this years ago in Africa and thought it might have been a rare occurrence. And at this stage, there have been many, many cases of adult worms still carrying a burden of these parasites. So this is a case, this is a small intestine that's been opened in an 18-month-old foal that had a huge infection, and therefore we removed ascarids up several thousand in this particular animal. Remember, when they're allowed to, they get up to a foot long, but like fish in a pond, they will be smaller if there were many, many. But this animal died of obstruction and perforation to the intestinal tract, the small intestine. Okay, we're going to talk about the strongvalves now, large and small. And they, in their migration, as far as their life cycle goes, to both small and large intestine, arteries, flank, and liver. And we again begin with infection on grazing. The larvae are on the herbage and are ingested, and inside our donkey host, they will complete their cycle by migrating into the intestinal wall or migrating into the intestinal arteries and other areas of the body. And then eventually they will come back to the large intestine. Male, female will lay eggs, passing feces, and they will develop to larval stages. So this is the stage we're interested in finding in this very large group. Why is this group important? Well, number one, the larval stages are ingested during grazing. The number of infected larvae will depend on the infection in the animals, will depend on how much grazing, time of year. But the strongyles, large and small, are important because there's more than 80 species. They're an enormous group. And we are grouping them for discussion, for teaching vet students, for helping people understand. We group them all together in one name. But the small strongyles alone are more than 50 species. Do we know their biologies? No. We do know some of the problems they cause. So, for the small strongyles, and you'll hear me use the word interchangeably with the cyathostomins, in an animal that was infected with the cyathostomins in the L4 stage, which is our encysted gut wall stage, every single one of those freckles is an encysted L4 cyathostomin. If we look at a higher magnification, we begin to see something's going on. Those look like larval worm stages. And when we go even higher, we see that those are individual cyathostomas encysted in the gut wall. Why is that important? Because this entire life cycle, even though all of these donkey parasites I'm talking about have long life cycles. What's long? The cyathostomas have up to about 4 months, and then they encyst in addition in the gut wall for up to 2 years. And we don't have diagnostic tools yet that can tell us what's going on in the gut wall. So we have to wait and be very vigilant about the fact that we have strongyl infection or strongyl levels on our farm or on our premise, wherever we're keeping our donkeys. So, what does this mean? If we look at the intestinal tract that's laid out of an equid that died of larval cyathostominosis, what that means is that all those freckles that you saw, all those individual larval stages, will emerge at once at certain times of year. We see it in late spring, early winter. We will see an emergence, and this has led all the red area. It was a hemorrhagic death in this animal because of all those parasites emerging at once. Kind of like insects do, where they get a signal and it's time to emerge. Spring has come. They come out of diapause. The other damage we can see is where one of the strongyles, a large strongyle that's called Strongylus vulgaris, but that large strongyle will migrate through the intestinal arteries that supply blood to the intestinal tract. And you can see I'm holding an arterial wall here. Now we're on the outside of the intestinal tract. But this is normal tissue, this arterial wall that I'm holding. And if you follow it along, you can see that this side of the artery is probably 30 times the thickness. And that is the response of the parasite or the response of the host to the presence of the parasite. What we're going to do in this picture is have a look inside that artery. Go back one. We're going to look inside the artery, and let's have a look at normal arterial tissue. You can notice how smooth and healthy it looks here. And as we move down into the arteries, craniomesenteric, ileocolic arteries that lead, carry blood to the entire intestinal tract, you'll notice here, if you look carefully, there, and there, those are migrating large strongyles. And they will spend about 4 months migrating in these arteries before returning to the intestinal tract to develop to adults, find their mates, lay eggs, and perpetuate the species. The problem is that this very thickened tissue that you see can break off and go down into the smaller vessels and lead to blockages, which can lead to infarct and death. So that's just another example of the strong bile. When they come back to the large intestine, they will develop to several inch long parasites. Those are the adults. The eggs are oval in shape, and we will spend time looking at these in lab for those who have signed up. Well, I'll mention that at the end, but the lab handout that has been made available to you, whether you make it to the lab or have time, and that has been, it's really a one-stop lab manual, which gives you almost everything you need. Based on the feedback we had, we thought, well, let's pull everything together. Why do you want to identify fecal egg count tests? What is it that you need to do those yourself, etc.? And I'll come back to that. The final group of detectable parasites are our tapeworms. And they invade or live and migrate in the small and large intestine. And let's look at that life cycle. And let's begin at the tail end this time. You're in charge today. Okay, that means, yes, right, but then I'm tethered to this. Okay, we want them to be in there, so I'll cooperate. Oh, that's okay. Oh, there you go. Eric, you're strong. Thank you. Okay, so let me take a step back. Okay, we're in the life cycle. Sorry, webinar people. I hope you're okay. Thanks for giving us that feedback. Okay, so let's look at the life cycle of the tapeworm. So the proglottids of the tapeworm will pass in the feces, and that segment of the tapeworm will have many eggs. What happens next is it's ingested by a grain or forage mite. And these are mites that live normally on the herbage, on pastures. And what happens is once this mite ingests that egg, there will be a development to a larval stage of the tapeworm inside this mite, and it's ingested during grazing, and the life cycle is completed. So all these parasites we're talking about today are grazing, are ingested by our donkey during grazing. Why are the tapeworms important? The jury seems to be always on and off, but the American Association of Equine Practitioners, which is the guideline which is in your handout, at this stage take the stand, and they are a guiding light for all of us, is that they do cause colic in equids. And we don't fully understand the extent, but at this stage that is what we all agree on. Inside an animal where we're aware that tapeworms are present, this is like 50 tapeworms attached to the junction between the ileum, the small and large intestine. So they can be present and are present in large numbers. And this is the kind of image and the clinical disease we follow to try to understand what impact they make. These are typical tapeworms, and every one of those little breaks is a segment. And that segment of the tapeworm, which is contains many, many tapeworm eggs. Okay, so what parasites can we diagnose? There's lots of detectable ones. We've only hit the top of the charts because of the time component. But if we look at a fecal examination, we see immediately our strong bile eggs and our asteroid eggs. And those are two of the most important and the most common. This image is in your handouts, and this is a table of all of the common helminth and protozoan parasites that occur in the equid. And what's circled are some of the strong bile. Okay, so in the diagnosis, okay. The test that we use that can commonly detect astrids and strongyles is the quantitative fecal flotation modified McMaster test. And a lot of times we just say the McMaster test. And this test is used, this test takes a fresh fecal sample, 4 grams. It uses a flotation medium. The recipe we're using in the lab and that you have in your books is a salt solution made from salt that you have in the kitchen. And we take that mixture and we apply it for that one animal, one donkey, and we count the eggs. And if we look at the number of eggs, we look at the okay, we are going to count the numbers of eggs under these grids, and we will spend more time in lab on that one. Okay. What are the best practices for deleting parasites? Let's take a step back. You've now determined you have strongyles, and you are probably, it would be a great opportunity at some stage for somebody to survey within this group what practices are being used. But the way that we want to manage, we love to delete them, sometimes we say that, but truthfully, the balance between parasites and their hosts, and I will share a story with you at the end, there's a lot of wisdom in managing and never thinking we're ever going to get ahead of their curve. Parasites are smart and they're going to outwit us. So when we say deleting, that gets your attention, but we'll probably always just be managing them. What are some of those best practice parasite management steps? Well, first of all, it needs to be practical. If removal of feces is not working for your operation, where you keep your donkeys, you're never going to do it. It may not talk, but for other owners, in my experience, it does. It must be affordable, must be available, and must be appropriate. And that usually comes down to your decision. And if we look at the large and small cyatostome, again, the strongyle life cycle, and we look at the bottom outside the host, so we're looking at 2 parts of this life cycle we can intervene, and that would be the environmental side and the host side. So let's have a look at some of the steps you may already be taking, may not be practical for you, but are a general picture of what can and is done. Are people here using a tool of anything like this to remove? Okay. A mechanical or human?
No, well, both.
Okay.
Actually, just purchase what you have here.
Okay. I'm going to show you the results of a study that we did where no matter how many drugs we have, sometimes just breaking a cycle, and our results have shown is very, very powerful. There are now a number of commercial vacuum cleaners available for the small operations. Manual fecal removal is done in some parts of the world, and in countries like Ethiopia and others, this bag of feces from the donkey will bring up to $2, and that's a gold mine for the poor owner. But manual fecal removal is still an option. You can rotate animal species. If you have donkeys grazing, but you bring in goats or cattle, and they come in, they're going to pick up the donkey parasites. They're dead-end hosts. You can in other words, all other hosts are not going to become infected with donkey parasites. You can also rotate your pastures if that's practical. You can leave land, if it's fenced and it's off, and leave it fallow for a couple of months, it eventually becomes safe pasture. Depends, 2 to 3 months, depends on how long it's practical for your operation. In some parts of the world, this fecal material from cattle and donkeys is made into plaster and decorated, and it's also used for flooring. Don't think this is a culture, I don't believe we do this in this country, but I'm sharing with you because it's extraordinary how practical the donkey feces can be. And I just thought of Fiona and her archaeo work, and I'm sure that's going to get her we'll be talking about that, I'm sure. Creating healthy compost. How many of you have compost in your operations? So you understand healthy compost, and you understand that there are all of the larval stages, fly, parasite, strong guiles. When they're brought into this healthy compost, they're often going to be ingested by other organisms that live. So it's a type of biological control. Beetles, for example, that are in the compost are going to feed on those parasitic stages. And then, of course, the significance, economic significance of a healthy compost is important for, it's beneficial in some way, either to you directly or by selling off to those interested. So I want to share with you a study that we did. One of my PhD students completed in South Africa, Sonia Matia, and she looked at best practice strategies in working donkeys. So she had a study on the veterinary school campus where she looked at the control animals, monthly fecal removal. She looked at pre-winter moxidectin treatment. The philosophy there is to give the best remedy available. At that stage, it was moxidectin in that country. To use it before the animal is going into a stressful period of time, where you want to get rid of all the parasites that you can. And then she combined the fecal removal with pre-winter. And she looked at, and there's a lot more data, but I sifted it out, brought into fine focus the nematode egg count, which is the tool that we're going to be looking at in lab. And that some of you, I'm aware, are already using. I've had some of those discussions you shared with me. And then the total helminths you're also seeing. So it was these two lines are the egg counts, and these two lines are the helminths. And when you compare these counts, they're dramatically reduced from the control animals in just a monthly fecal removal. The treatment of remedy was very strong, but the two together was also stronger. So once again, one has to determine what works in your situation and then test it. And the testing part you can do with egg counts. I wanted to share with you, have you folks heard of the nematode trapping fungi? Okay, and you've heard about it, but at this stage, I wanted to share with you a little bit of the background. This is an infective L3 strong guile, and these are the fungi that are trapping it. Some years ago, in our collaborations with our Danish colleagues, and these fungi are in the environment all over the world. They're not just in one country, they're everywhere. And these are stimulated to want to trap and kill and ingest these larval stages. So we've done, what the Danes were able to do were to culture the fungi, and then through various projects we were able to test the impact of these fungi in decreasing the numbers of infected larvae that would be available in the environment. And the studies have all been extremely successful. This was a few years ago, so some of you may say, well, what's the deal? Why didn't it get out there sooner? The commercialization took some time, finding a company that wanted to take up what was being grown and get it out there, and we also needed test studies to validate. And so that product, and this is another example of the trapping of these fungi. So the way it works is the spores are fed to animals. They don't hurt the animal at all. They pass in the feces, and in the feces, they will, as soon as the infected larval stages are in the environment, they will go after them and basically eradicate them. So this product is being launched in the near future, and it may become a tool that we will be using in this country to get through FDA. So that is a new innovative approach that we all welcome, because it has a great opportunity for reducing numbers. Okay, now we're looking at our life cycle of the strong guiles, and we're saying, how do we intervene? How do we break the life cycle, reduce the numbers of parasites by working with the host? Oh my goodness. Okay. Let me go back. Yep, doesn't want to go back. Sure. All right. Right there. Huh. Let's see. We're not finished. So if we go back to, maybe it's hungry. There we go. Let's try and see. Okay, so what do we know about the remedies that are effective in our, what remedies are available in the U.S.? And again, this comes out of the AAP guideline, this table. And many of you are familiar with fenbendazole, which is the benzimidazoles, the pyrantyl products, and of course our macrolides, our moxidectin, ivermectin. And those three groups of products are available in the U.S. and labeled for horses. And we do use them in donkeys, but we are extrapolating. The next table, same table, but I want to bring other information, is the question of drug resistance. The escalation of the development of resistance by these parasites I'm talking to you about today, and how quickly they have built resistance, has been frightening. I never, ever thought we would ever stand here telling you that Parasites eporum, and we would have resistance to the drugs we use to treat what is one Parasites. There's two Parasites, but basically we talk about eporum, but there's a second one that's been reported. The point is that we never thought we'd be, and this says that we have early indications of resistance to the benzimidazoles, early to the pyrimidine, and widespread to macrolides. The other information in the table to bring to your attention are the small strongyles, the cytostomas, widespread all the way across the groups. And the large strongyles, though it says none, we have had indications, early indications of resistance. So we have there's the length of time that these drugs will still work is limited, and that's the point of this slide. So let's look at our basket of best practices. We've talked about several number of best practices. We've talked about pasture hygiene, knowing what information, the available information available of what do you need for your donkey project and program, the parasiticides that are available, what's available, what works. And remember, when we talk resistance, we don't mean, if we did a survey in this room, does not mean that every group of animals and those parasites are resistant. It's knowing, is it yes or no, and then building on, if you have low resistance, keep it, but it means managing well. Other basket components are management practices, integrating several methods together, as Sonia did with her study, rotational grazing, composting, and the last one is monitoring, which is absolutely what you can do by having either you or your veterinarian or you have excellent labs available to make sure there's monitoring regularly throughout the year in your farming system, your donkey management system. Okay, I want to share a quick story. How are we doing on time? For a few minutes. I want to tell you a story about the zebras, which in southern Africa you often hear us call them zebras, so I may slip back to my former the wording, but we always saw them as the striped thoroughbreds of Africa. What's extraordinary about the zebras across southern Africa, and those are the ones I've studied, is number one, they're free-ranging. Number two, they've never been treated. There's no intervention that is practiced on them as far as parasite control. We have, and number three, we've yet to see a zebra, they're very fat. They're very, very high, good body condition. They're not fat. Is Karen still with us? They're very, very healthy, but we never see clinical disease, and we all never see colic. And that applies to the animals that are free-ranging. However, they have, as we said, a very wide biodiversity, which they share with all their other equine cousins, as far as numbers of species of parasites that they live with. But we see in zebras burdens of parasites of hundreds of thousands of strongyles and millions to 100 million pinworms in individual animals. And that's a lot of the research I've published. And so that work is out there. That data is published. What's extraordinary is, and this came from, and what I want to share with you now is we worked in Southern Africa, and what I'm going to tell you refers back to about 80 to 100 animals that we studied across Namibia and South Africa. Different zebra species. And why I'm telling you this is this may be an opportunity for a new innovative tool, because with the resistance that we're looking at has taken place in the developed world, especially in countries like here, we're looking for new ways of control. So we go away from this point. This is a female worm. This is a microbial community associated with a female cyathostome in a zebra. This is another species, and the tail, this is the female tail. Same here, it's just a shorter, different species, a different tail size. We found that these were microbial communities associated with strongyles in a mammalian host. It was the first report ever. Today, it's still the only report in this particular, because nobody else seems to want to study the zebra. It took a lot of work to get the parasites out, but the microbial constituents that we found in that microbial community, we're looking at a cross-section. This is the vulvar opening. Why is that significant in the worm, in the nematode? In this cyathostome, that's where her eggs are laid, which are shed into the environment. And at that time, we looked at these microbes. They were long and segmented. These were round and segmented. These were septate, spirochete. There were 10 different groups. And at that time, we pulled together this extraordinary group of scientists. We tried to culture these organisms because we wanted to know, why are they there? Are they a type of biological control? Are they there because they have an association that's somehow managing these huge parasite levels? We wanted to know answers. We did everything we could to culture, characterize. We got down the road, published everything we had, and we left the story on the shelf. And I had a lot of material in freezers, and I still have very good contacts in Africa, so when zebra material comes available, we have that opportunity. Recently, we reopened this book, and we looked at this, and one of my colleagues is several team members. Today we have new tools. We have microbial genetics sequencing tools that will help us ask, who are they? What are they doing? What are their activities? So we have put the team back together. We've got new folks coming on, and I hope that we are doing a comparative study, zebras, donkeys, but we have to find those animals, and that's a challenge, that have not been treated ever. And I'm working through a lot of countries to try to find some of that other material. So where have we been? We've talked about what are the detectable parasites, what are the diagnostic, well, those that we can diagnose easily, and which ones can we delete or manage. Oh. I'm so glad you're over here to do this, because I just have 2 more slides. It's just okay, so here we go. I just wanted to share with you that this afternoon, and all of you have this handout if you're overbooked and not able to come into the lab, and I understand that there's only so many people that are coming in, but we will be in the Cole facility. This handout is your go-to one-stop handout to help you be practical in making a diagnosis. But if all else fails, and we can't get ahead of the curve of our parasites, or the drugs just aren't available, or we don't have the diagnostics, I want to share another alternative remedy with you. Markham's masterpiece, Gervais Markham wrote this book, The Masterpiece, that contains all the knowledge belonging to the Smith barrier or the horse leech, touching all curing of all diseases in horses. He wrote that 500 years ago. It was a bestseller. It had one edition after the other. Horse leech are the veterinarians, of course, and the rest of us are the Smiths and the barrier. So Markham was practical. He taught strong vowels, he taught thoughts, he taught aspirins, and he had lots of recipes to help us get rid of those parasites. So I'm going to leave you with this remedy, which is to give great slices of white bread, toasted at the fire, steeped in muscadine, pine nuts and wine, or else a spoonful of soap of brimstone milk. So don't give up hope. We'll get those parasites in control. I just want to end with thanking Eric, his team, the students, and a number of people who made material available, and Heather Moberly, who helped with publication search, and my husband Eddie, who I couldn't do it without the technical support. His is always there. Thank you very much. Healthy compost is compost that's at a high level. By the time, so I'm envisaging healthy compost that's going to, I always want to remember this, I want to say it's centigrade, but it's high enough that everything in there is at a high level. So the parasites would not thrive, or they would have been eaten by some of the succession planting insects that live in a healthy compost heap. So when I say healthy compost, it would be certainly a parasite-free material if done correctly. I mean,
do you think it's likely that there are any donkey populations, perhaps in Eastern Africa somewhere, that have never been, have never seen a dewormer? And I'm just wondering whether the parasites in those might be similar to the zebras, with those sorts of same.
Great question. Great question, and I'm hopeful that there may be populations of donkeys that have not had treatment. I don't know how, the homemade remedies, I'm just assuming, I'm talking modern intervention, and so I'm welcoming, I do have some donkey samples from South Africa we're going to take. We're beginning with this microbiome study. We have material from zebras and from donkeys, but I would be delighted if we knew, and the answer, be able to look at, number one, if the parasite burdens are similar, but the other is, of course, this whole microbiome story of what kind of relationship do these same parasites or similar have with these microbial communities. So I'm very interested to find, if we can find donkeys that are not treated. Fantastic.
BLM donkeys was our first project that were not treated.
My understanding is, I've been in contact with Al King, and my understanding is that somewhere along the line, probably in this country, those animals may have been somewhere. I don't know if we can guarantee, but it is a source that we are looking at. And once we get started on the material and what we're doing in the lab, we'll be certainly looking. Maybe that's an option we haven't totally finished looking at. We have great contacts, and it's appreciated.
I was on the compost comment. I was wondering how you felt about the fly predators that they saw in the mail when they hatch and all that.
The fly predators are a great example. my students always ask, and I think it's a great innovative approach. But at this moment in time, there is not enough hard research evidence that can tell me to say, put it on the list or not. But it's practical, appropriate for a lot of equine owners, and they swear by it. And some of my students will come and talk about it, say their family is using it. So I think that I think it's an option, and we just need to get some more hard data out to try to determine what impact it's making.
Maybe it helps, definitely doesn't hurt.
And that's what I've heard, it helps, but definitely, it may help, but it definitely doesn't hurt, right? So it's absolutely another, I think in time that will develop to a, maybe more precise, so you're getting a return for your money. And don't get me wrong, but I mean, that's what people ask. But I think it's a great tool. There's no webinar, people?
Question over there?
Yeah, you're gonna basically settle a family argument with this one?
This is terrific information. I mean, please take it to heart.